As you know, in recent years, there has been an increase in interest in using plant extract in livestock production, and this is for different reasons. The first one is that the feed industry or the livestock industry has been always seeking for innovative technologies to improve feed efficiency and also to reduce the impact of livestock production on the environment. Second, there is an increasing pressure from the society or the consumers to reduce the use of feed antibiotics and growth promoters in livestock production because of the possibility of the emergence of antibiotic-resistant bacteria. Finally, because plant extracts such as essential oils are perceived as natural and safe, and therefore they are more accepted by the consumers. You know, plants produce an extensive array of organic compounds that are synthesized through their secondary metabolites. And among them, we have tannins, saponins, and essential oils. And the latter, when extracted from the plant and concentrated, they can be used in animal nutrition for their aromatic and more particularly for their antimicrobial properties. How can we define essential oils? They are the volatile components extracted from plants by different methods of distillation, mainly steam distillation. They can be extracted from many parts of the plants, can be roots, bark, or flowers. And contrary to what their name may suggest, they're not essential for nutrition or metabolism of the plant, nor are they oils in the sense of being lipids. They're responsible for the fragrance and the flavor of the plants. Chemically, we can define essential oils as complex mixture of alcohol, ester, or aldehyde derivatives of phenylpropanoids or terpenoids, with the most abundant terpenoid essential oils being the monoterpenes. These two groups, namely terpenoids and phenylpropanoids, are synthesized through separate metabolic pathways. For terpenoids, they can be derived from the deoxycellulose and the mevalonate metabolic pathway, while the phenylpropanoids, they're derived from the shikimate metabolic pathway. This here, example of this compound are sanamaldehyde and eugenol, whereas for terpenoids, they can be either monoterpenes, such as thymol or cavacror, or sesquiterpenes, such as gossypol. Given the diversity in terms of chemical composition of essential oils, it is unlikely that their antibacterial activity is due to a single mode of actions. We know that essential oils are lipophilic, and therefore they're able to accumulate in the lipid bilayer of the plasma membrane and disrupt the cell wall structure. This mode of action is typical of terpenes. They can disrupt the ion gradients across the plasma membrane, which leads to the dissipation of the ATP pool within the cell, which can limit the growth or even cause the death of the cell. This is typical of the mode of action of phenolic compounds such as cavacrol. Finally, they can interact with proteins either within the plasma membrane or within the cytoplasm itself, and this is the case for sanamaldehyde, for example. You know, as I mentioned earlier, there is a wide range of essential oils, and we can understand that researchers have heavily relied on in vitro approach, short-term in vitro approach, to screen the large number of compounds. They use different techniques, pure cultures, batch culture, incubation for 24 to 48 hours, or continuous culture system, which means incubation for up to 8 days. However, we should keep in mind that the true potential of essential oils to improve feed efficiency, and stress here protein and energy efficiency, and by the way, ruminant performance should be assessed in vitro, in vivo, in long and term experiment. So the objective with using essential oils is to improve efficiency, as I mentioned earlier, of protein and energy utilization in ruminants. So let's start first with protein metabolism. As you know, the protein metabolism in the rumen is a complex process that involves different steps. This is the catabolism of protein. You have proteolysis, the first step, which means the degradation of protein into peptides. Peptidolysis, which means the degradation of peptides into amino acids. And the last step, which is the deamination, which degrade or ferment amino acids into ammonia. And this catabolism is an inefficient way to use protein, and that makes ruminants among an inefficient user of dietary protein. For example, efficiency of transfer of feed nitrogen into milk averages 25%, as the remaining being lost, being excreted with urine and feces in the environment. So, if we'd like to use essential oils to improve efficiency of protein utilization, essential oils would result in a reduction of protein degradation and also ammonia production. So on this slide, I'm reporting an in vitro results from an in vitro study by Newbold and R 2004, in which they investigated the effect of a mixture of essential oil composed of thymol, eugenol, vanillin, and limonene, and they investigate the effect of this mixture on protein, ruminal protein degradation. As you can see, among the subsequent steps involved in protein degradation, only the last one, the deamination activity, was reduced by the mixture of essential oils. The same observation was confirmed by McIntosh and al., who reported a slight but significant decrease in the rate of ammonia produced from casein acid hydrolysate. And this decrease, if you see here, they did not observe further decrease in ammonia production when essential oils, the mixture of essential oils, was combined with monansen. These results suggest that the mixture of essential oils inhibit the same group of bacteria that is sensitive to monansen, which is gram-positive bacteria, and this group of bacteria is called hyperammonia producing bacteria, which are present in low numbers in rumen, but have a very high activity of deamination. And actually, this observation were confirmed by the same authors using pure culture of ruminant bacteria. And as you can see here, the mixture of essential oils was able to inhibit the growth of some representative of the hyperammonia producing bacteria. And the concentration required to inhibit that growth was about 40 ppm, which is higher than that likely to be achieved in vivo, as we will see later in the presentation. So on this slide, I'm reporting the effect of, this is by Busky and al., the effect of different essential oils on ammonia and VFA concentration. As you can see here, the response is dose. There is a dose effect of essential oils, and higher dose of cinnamaldehyde carvacrol eugenol, 300 and 3000, were required to inhibit ammonia production in vitro. However, as you can see here, total VFA production was also inhibited, which is a reflection of an inhibition of the degradation of feed in the rumen. And this means that less VFA are produced, and of course, this is something that we don't want it to happen. We don't want it to happen in ruminant, because VFA are the main source of energy to the animal. These results are from Castillejos and al. 2006, where they investigate the effect of thymol on protein metabolism using 24-hour batch culture. And as you can see here, at high doses, 500 and 5,000 milligram per liter, ammonia production and branching chain VFA production were reduced, which is an indication again of deamination of ruminal activity by rumen microbes. However, when the same compound was evaluated in continuous culture system, which means 8 days of incubation compared to 24 hours for batch culture incubation, ammonia production was not affected. However, as you can see here, there is an increase in large peptides and small peptides plus amino acid concentration, which means that the effect of the mixture of essential oils was mainly on proteolysis and peptidolysis. As I mentioned earlier, most of the studies conducted today with essential oils are really laboratory-based and of short-term effect. And here, there are some in vivo studies that were conducted using different types of essential oils. As you can see here, the results in vivo, it means in live animals, did not confirm the positive effect observed in vitro when using essential oils. And this is more likely because of the different dosage level used in vitro compared to in vivo. As you can see here, it ranges from 10 to 26, except these studies where the concentration, the doses used were excessively high. Anyway, there is no effect on ruminant ammonia concentration, which means protein degradation was not affected by a different type of essential oils. Now, if we move to the potential to use essential oils to affect positively energy metabolism in the rumen. Before going into the details of the results, just very quick reminder about energy metabolism in the rumen. As you know, the feed, once ingested by the animals, it goes under an intense degradation fermentation in the rumen, which result in the production of volatile fatty acids. This is the main source of energy again to the animal, mainly acetate, propionate, and butyrate, and also results in the production of methane, which is a loss of energy. And just to mention that fermentation patterns which reduce acetate production and increase propionate production will decrease, in theory, of course, methane production in the rumen. So essential oil can be beneficial to improve efficiency of energy utilization if they increase propionate production, they decrease acetate and methane production, of course, without decreasing total VFA production. On this slide, it's from Machbeuf and al. From INRA in France. They investigate the effect of thymol and Thymol oil at the concentration of 300 milligram per liter. As you can see, both thymol and Thymol oil, they decreased methane production in vitro. This is 16-hour batch culture, but at the same time, again, VFA production was also inhibited. And if you see here, the extent of decrease in methane and VFA was higher with Thymol oil compared to thymol, which means that other constituents in the Thymol oil contribute to the antimicrobial activity of Thymol oil, and this is particularly the case for P-cymene. This is a slide representing data from Bousquet et al. Again, where they investigate the effect of garlic oil and its derivative, which is the diallyl disulfide, used at 300 milligram per liter in 17-hour batch culture. And as you can see here, both compounds here, they dramatically reduced methane production in vitro, but this was associated again with strong decrease in VFA concentration and strong decrease moderate decrease, in dry matter digestibility. And as you can see here, this derivative DAD, the extent of decrease in methane was almost the same as with garlic oil, which means that the sulfur compound is responsible for most of the antimethanogenic activity of garlic oil. This is another slide from the same authors, where they investigate again the effect of garlic and DAD at 300 milligram per liter. In first experiment, they used batch culture, 24 hours. Second experiment, they used continuous culture, 8 days of incubation. And in batch culture, as you can see, both compounds decreased VFA production. However, when the same compounds were tested at the same concentration in continuous culture system, 8 days of incubation, no effect was observed, neither on VFA nor the dry matter digestibility. This means there is a possibility that the microbes adapted to garlic and its derivative DAD, or even they degraded these compounds. And this is actually what explained the lack of effects observed when these compounds were tested in vivo. Here you have a slide from Kloeven-Hussen and al. In sheep, of course, and they fed high doses of garlic and DAD, 5 gram and 2 gram per day, and nothing happened on methane production or dry matter intake or digestibility or VFA concentration. The same observation was also observed by a group from Wageningen University, where they tested at 2 different doses, DAD, 56 milligram per day per kilogram and 200 milligram per kilogram, and again no effect was observed here on methane production. So other in vivo studies have evaluated the effect of different essential oils ranging from garlic to juniper berry essential oil to cinnamaldehyde or oregano essential oil. And as you can see, for different feeding rates, no effect was observed on methane production. And this discrepancy between in vitro and in vivo studies, they are probably due to the fact, again, that the doses that have inhibited methane production in vitro are too high, again, to be achieved in vivo. And if you take this study by Boschman and McGinn from Lethbridge Research Center in Canada, they fed beef cattle, again, with the same essential oil mixture at the rate of almost 1 gram per day. And as you can see, the concentration reached, calculated, of course, immediately after feeding, would be around 33 milligram per liter. If you take another experiment, which was, sorry, not this, yes, of course, which was run by McIntosh and al., who investigated the same essential oil mixture for its effect on one methanogens, Methanobrevibacter smithii. And as you can see here, the inhibition of these methanogen occurs only when the concentration of essential oil in the medium is about 1,000 ppm. 1,000 ppm, sorry. So let's move to the effect of essential oils on ruminant performance. And although there are more and more studies trying to evaluate the effects of essential oils in vivo on rumen fermentation and diet utilization, surprising, there is a few studies that have investigated the effect on ruminant performance. And I summarize some of those studies here. Again, this is on dairy cows. And as you can see, for different types of essential oils, whether a mixture or two essential oils combined, you can see here that, again, the results were disappointing because no effect was observed on dairy cow performance. And the same story happened exactly with gain in sheep and beef cattle, except this study where we observed an improvement in feed efficiency. No effect was really, we cannot say that from this slide that there is clear effect, positive effect of essential oil on ruminant performance. So there is one thing that was really a little explored, is the fate of essential oils in the gastrointestinal tract of the animal, particularly the ruminants. And this study is from Malik and al., again from INRA, where they evaluate the effect, the ruminal disappearance as a percentage of the dose fed of monoterpenes in the rumen of dairy goats fed low level or high level of these monoterpenes. As you can see here, the high of the ruminal disappearance, it was almost 100%. It means that 100% of the terpenes fed to the animals disappeared in the rumen. So different mechanism action can be used to explain the lack of effect of the disappearance of terpenes from the rumen. First, the bioconversion of terpenes by rumen microflora. that have been shown in different studies. The transfer to the gas phase of the rumen due to the volatility of terpenes, favoring losses during rumination and eructation, and the absorption of terpenes across the gastrointestinal tract into the blood system and excretion in the urine, and therefore they can be transferred. Once absorbed, they can be transferred to animal products such as milk, for example, which may potentially affect the organoleptic properties. So to conclude my presentation, I would say that today, up to now, most of the antimicrobial activity of essential oil was mainly demonstrated in vitro, which again is a short-term effect. When effective in vitro, positive effects such as a reduction in protein degradation or in methane production were counterbalanced by an overall inhibition of rumen microbial fermentation. Many of the concentration, again, that have resulted in favorable response in vitro are simply too high for in vivo application, which also can lead to palatability issue in ruminants. The favorable effect observed in vitro may not be, again, as marked as in vivo because of the adaptation of the microbes to essential oils and or the degradation of essential oils by rumen microbes. And this is actually what explains the discrepancy between in vivo long-term and in vitro short-term experiments. When used at practical feeding rates, as shown in different slides, small or no effects on rumen fermentation and rumen performance. And finally, further in vivo studies are needed to assess the real potential of essential oils as feed additives in ruminant nutrition. And with this, I would like to thank you for your attention.